guys?
Like,
Lactic acid,
Yes,
okay?
that's what it is.
And lactic acid is acidic. So the immediate problem is too much CO2 buildup, not enough oxygen to function. Over time, obviously, if those cells are deprived of oxygen, they're going to be breaking down. They're still going to be doing metabolic activities, and they will end up building up lactic acid. And of course, lactic acid can cause muscle fatigue, muscle soreness, and that's just the muscles. That also affects everybody else. This is why a lot of times after somebody has stopped breathing, when they restore their breathing, they bring them back using CPR. CPR is your cure for acute respiratory acidosis. They want to take you to the hospital. One, because when you do CPR correctly, you probably have broken a rib, so they want to make sure everything's set properly. But two, they're going to monitor you because that lack of oxygen, building up of lactic acid leads directly into metabolic acidosis. So this is why sometimes if somebody has a bout of acute respiratory acidosis, they stop breathing, the next day they may talk about how tired they are, how sore they are. That's the metabolic acidosis lingering after we've restored breathing to them. So CPR is what we use to counteract to help obviously restore those actions. When you do CPR, the breathing in their mouth gets all the glory because that does put oxygen back into the lungs. But the chest compressions are the most important because they are going to mimic the respiratory pump. That is going to allow that CO2 to be forced out. That's going to allow the blood to continue to circulate, and that respiratory pump activity again gets rid of the CO2 until the patient's able to start breathing again. Okay. Chronic respiratory acidosis, you're breathing, but you're not breathing as much as you should. So you have chronic low levels of breathing. So there's some kind of impairment going on that, again, we haven't completely failed yet. We're still doing it. We're just not doing it enough. So hypoventilation can be due to central nervous system injury. So somebody has a brain injury to the medulla, to the pons, and it changes their breathing rates to where it's so low it causes issues. This can happen if it's desensitized by drugs, alcohol. Again, when you release alcohol in the system, the brain is going to release GABA, right, or gamma-butyric acid. We learned that in A&P1. And GABA is an inhibitory neurotransmitter. It can help with anxiety, which is why some people drink to get rid of their nervousness. But when you release GABA, it slows down neural firing. That's what makes the anxiety go away. And of course, if we slow down neural firing in the medulla, we slow down breathing rates. So if you are an alcoholic, you can end up with chronic respiratory acidosis because you're just suppressing your breathing for so long. If you are taking barbiturates, again, downers, that's why they're called downers. They're going to, again, decrease the action potentials coming from the medulla, slowing your breathing rates. So again, drug addicts may develop chronic respiratory acidosis. I know it's not really high up on their worry scale because, all the other things are happening, but still not a good thing. And of course, this is why you can't take pills and alcohol at the same time. You get one downer, you get another downer, double down, you stop firing. And of course, if the medulla stops firing, you stop breathing, and chronic because it becomes acute respiratory acidosis really fast. And that's why that combination of alcohol and pills is almost immediately fatal to just about every famous person who's done it, right? So many people. Don't do drugs, kids. Just saying. Okay. If the brain is okay, there's probably a problem getting air into the lungs. So this is where COPD comes into play. So when you have emphysema, again, it causes a saturation of CO2 in the air in the lungs. And of course, if we look at the air in the lungs, if it has the same or higher amounts of CO2 than the blood, then of course we're not going to have diffusion rates happen like they're supposed to. So that air is preventing the diffusion of CO2 out, and it builds up in the system. Pneumonia can cause this. This is why a lot of times after people get over pneumonia, they feel tired for quite a while. It's because they've developed this issue and they have to work through it. Any kind of blockages going to the lungs, stuff like that. So whenever you have COPD, one of the things, again, they're always monitoring is your oxygen saturation levels, but that in turn is also going to help you predict your CO2 saturation levels. So they want to make sure you don't have too little oxygen or too much CO2. And of course, putting you on that oxygen tank with the COPD is going to help remedy both of those issues. Because if you saturate the air in your lungs with oxygen, then there's not a lot of CO2, so CO2 would diffuse out at normal rates. Okay. So here's just showing you our problem. We have hypoventilation causing a buildup of carbon dioxide, which is acidic. So that is going to lead to respiratory acidosis. What we do to fix that, obviously the brain is going to say, hey man, you should speed up your breathing rate. So we're going to increase respiratory rates. This is why a lot of times you'll see people with COPD pant. They're having trouble getting that oxygen and carbon dioxide to exchange properly, so it increases their resting breathing rate sometimes, which may or may not be beneficial for them. And of course, the kidneys are going to kick in to try to help out, to get rid of some of those extra acids to bring us back down. And of course, we see this in non-life-threatening issues. You're exercising. What do you do when you exercise? You increase breathing rates because you're making more CO2 and using more oxygen. So whenever you're exercising, we increase breathing, which is why we start panting by the time we get to the top of the stairs coming to class in the morning. Okay, so you've had a tiny amount, a little nibble into what respiratory acidosis is like. So you've increased breathing to get rid of that CO2. Respiratory alkalosis, obviously the opposite of acidosis. This is your third most common type, okay? Bless you. So it's relatively uncommon. It usually fixes itself before it causes major problems. So it's due to hypocapnia, too little CO2. So again, too much or too little of anything can be problematic. So even though carbon dioxide is a waste, we have to maintain the proper level of that waste to maintain the pH of the bloodstream. So this is due to hyperventilation, breathing too much. Seldom persists long enough before to cause clinical emergencies or anything like that. Typically people pass out and then the problem's solved, right? Because when you sleep, the problems go away. Okay, so causes physical or psychological stresses. So again, you have an anxiety attack. It causes you to increase your breathing rates. As you increase your breathing rates, you oftentimes expel more CO2 than you need to expel, and that causes you to drop those CO2 levels, shifting the pH of your blood, shifting the pH of your cerebrospinal fluid, and when your CSF gets off on the pH, it makes the neurons go to sleep and you pass out. And magically, once you pass out, whatever was stressing you goes away because you're asleep and your breathing returns to normal. So you will wake up not long after the breathing goes back to normal, the levels go back to normal. And again, this is why they tell you to breathe into a paper sack. Again, paper sack, not plastic sack. That'll kind of end all your problems really fast. So don't do that one. But of course, when you're breathing into the paper sack, that saturates the air in the sack, thus the air in your lungs with CO2 that prevents it from leaving so quickly. So that hopefully will prevent you from that passing out issue. And again, that's when someone's going to be there calming you down, that kind of scenario. Moving to high altitudes, because as we learned, the higher in altitude we are, the less oxygen there is. And of course, if we don't have enough oxygen in our bloodstream, that's going to kick in those mechanisms in the respiratory areas to speed up breathing to get us more oxygen. Unfortunately, it gets us more oxygen, but it gets rid of that CO2 faster, which can cause issues. This is one of the reasons why when you go hiking in high altitudes, so like if you go to Colorado, go hiking in the mountains, you might feel lightheaded while you're hiking. A lot of people attribute that to, it's altitude sickness. Yes, and altitude sickness in one part is oxygen issues, but the bigger part of altitude sickness is CO2 issues. So you're panting because you're hiking and you're in higher altitudes, so you're getting rid of CO2 too quickly. You're on the verge of passing out because your CSF is going to be a little bit more alkaline. So if you're feeling lightheaded while you're hiking, that's just code for about to pass out. Breathe into a paper sack or sit down, calm your breathing, relax a little bit, that scenario. So moving up there promotes hyperventilation, which is why you see so many people pass out at high altitudes. It's not the lack of oxygen that usually causes you to pass out. It's the excess or the lack of CO2 because you've got rid of that too much. If you're on a mechanical respirator, so this is, again, one of the reasons why they pay the respiratory therapists the big bucks. They have to set the machine at the right level because if they set it too high or too low, if your breathing is too high or too low, they can cause acidosis or alkalosis. So they have to keep the breathing rates just right so the pH stays just right, which is why it always beeps on you, which is kind of mildly annoying. If you work in the ICUs, there's all kinds of beeping machines. And then you'll often hear people like, I wish the beeping would just stop. But not really, because that means the patient's dead. But it can get on your nerves. But that beeping, again, is indicating breathing rates that everything's just the way it's supposed to, not too much, not too little. And of course, brainstem injuries. Just like damage to your medulla can cause your breathing rates to slow down, causing acidosis, they're just as likely to cause it to speed up and cause alkalosis. It just depends on which way your brain swings, if which condition is going to cause you. And this essentially is the opposite picture from what we just saw. If we get respiratory alkalosis, we're going to pick that up. So we're going to have decreased respiratory rates coming in from the medulla. And then we're going to, again, secrete out the bicarbonate ions, retain some of those hydrogens to bring our acidity levels back up. Metabolic acidosis, the second most common type of disturbance that you're looking at. So causes, of course, too many acids. So how can we get too many acids? One, we've made them. We've done something or created a condition where acids build up metabolically. So, of course, we have lactic acidosis. So we see lactic acidosis after strenuous exercise. So that is a case of lactic acidosis in the muscles. So you get the tiredness, you get the fatigue. This may take a few days to cycle through the system and get rid of those acids. The liver's going to kick in doing that Cori cycle and it'll clean up that mess. Or prolonged tissue hypoxia. Again, if we stop breathing, the immediate problem is respiratory acidosis. But when we stop breathing, we build up lactic acid, which will then kick us into lactic acidosis. So this is something that you're having to be monitored for if you have those issues. And then, of course, ketoacidosis. This is the type of acidosis some people pay money to be put on, right? You go on the keto diet, and when you're in the keto diet, you maintain the proper level of ketoacidosis. Too high and you die, too low and you're not burning fats properly. You got to be just right with the level. Which is why they constantly do P testing if you're doing your keto diet properly. They want to make sure your acid levels in your urine are where they're supposed to be, not too high because they don't want to kill you because that is bad for their business, and then not too low because then you're not going to lose weight, which is bad for their business. Okay? So too many keto bodies, fat breakdown, and if you're not exercising while you're burning the fat, the keto bodies build up, causing ketoacidosis. And again, this is why some people, when they go on the keto diet, if they're not doing anything other than just this diet, they're not increasing exercise or anything like that, they'll complain for like the first 2 weeks of getting the keto flu, where they just feel really tired and sluggish and aching. They don't feel good. It's like, it's your body saying, I'm dying, stop it, feed me a freaking donut. But unfortunately, again, this is what they're going for. So if your liver's okay, you'll pull out of it. If your liver's not okay or you do it a little bit too much, you may not pull out of it. Which is why they always have the disclaimer, please talk to your doctor before changing your diet drastically. That's so you don't sue them when you have problems. Now, if you're not making excess acids, you haven't changed your diet drastically, you're not starving yourself, you're not exercising strenuously or holding your breath too long, you can have issues with the kidneys, of course, because this is metabolic acidosis. If those kidneys aren't able to get rid of the hydrogens, or if sodium reabsorption stops. So that's the tricky part. When we bring sodium back in, we kick out potassium, and when we kick out potassium, hydrogen is going to follow. So if we stop bringing in sodium, we stop kicking out potassium, and we stop kicking out hydrogen. So that can lead to that acidosis issues, and of course it can lead to excessive amounts of potassium, which is what we saw yesterday when we looked at if you have too much potassium, you often get acidosis because hydrogen's linked and hangs out with that potassium. So even though everyone's like, oh, sodium's so bad, you got to have those right levels. So if you don't make enough aldosterone, if you don't bring in that sodium, that can cause issues. And of course, severe bicarbonate loss, chronic diarrhea, because again, we are going to drop off bicarbonates at the digestive system and hopefully reclaim them so they can cycle back around and continue to absorb acids. If you're having diarrhea, you're pooping before you can reabsorb the bicarbonates and the electrolytes and the water and all those issues. So when you have diarrhea for several days in a row, a lot of times patients complain about how tired they are, how weak they feel, and a lot of them attribute it to, oh, I'm dehydrated because of the diarrhea. It's like, you've got metabolic acidosis because of the diarrhea. So you got to be careful of that because it's going to throw off most of your issues. So having diarrhea for too long is not good for your body, both for water purposes as well as for acid-base balance purposes. And then metabolic alkalosis, number 4 on the list, because you can't get any lower because there's no more. So this is the rarest one to get. Here we have too many bicarbonate ions in the system. So some causes: vomiting, Okay, so when you vomit, you're going to get rid of your gastric content. And again, gastric content is very acidic. So what happens is your stomach will make the gastric secretions to replace what you've just lost. And your body perceives, oh, we're making acids. I need to make some bicarbs to counteract those acids. You vomit again, you get rid of those acids, so you make new acids. And your body's like, oh, we're making acids, let's make bicarbs. So every time you vomit, you replace the acidity levels that you've lost, but your body makes more bicarbs because it doesn't realize that you've lost the acid. So it's smart but stupid. So every time you vomit, you double down on those bicarb ions. And more bicarbonate ions, the more we absorb acids in the blood, and we end up with metabolic alkalosis. This is what happens if you have bulimia. So if you're anorexic, you can have ketoacidosis from lack of nutrients. If you're bulimic and you vomit all the time, you can have metabolic alkalosis. And this is also one of the issues we have with patients who have to go through chemotherapy. If they're vomiting a lot with chemotherapy, they can feel really tired, really weak, and not feel good because it can kick in the metabolic alkalosis, which also affects their ability to respond to cancer treatments. So if you have a patient who is vomiting a whole lot, they will again give them that expensive medication to stop them from vomiting. So this is also why when you have the stomach flu and you just vomit for a few days in a row, it's like, why do I feel so tired? It's like metabolic alkalosis. Now, if you get the full flu, right, you vomit for 2 days and you diarrhea for 2 days, they'll balance each other out. But who wants to balance that badly? Okay, so you're going to feel like crap for a few days after big vomitings. You're going to feel like crap after a few days of crap because, again, one's going to send you to metabolic acidosis, one's going to send you to metabolic alkalosis. Okay, so it's just not going to make you work right. So treatment, stop vomiting. So again, this may require medication to do that if it's due to chemotherapy or a stomach bug or anything like that. And if it's really, really severe, they may have to administer ammonium chloride. So what happens when ammonium chloride hits the liver will metabolize it into hydrochloric acid, which at first glance doesn't sound great. Hydrochloric acid will eat through living tissue, and we don't want that floating around in our body. We contain it in the stomach. But again, at that point, if you are administered this, you have so many bicarbs floating around. As soon as the liver releases those small amounts of hydrochloric acid at the cell level, bicarbs absorb it almost instantly. So it's basically like, a little bomb to get rid of everybody just around it. I think of it like those UV bombs in the second Blade movie where all the vampires just poof, disappear as soon as the UV light hits them.
Yes.
Remember Blade 2?
Yes, I do.
If you don't, it's okay. Neither did Wesley Snipes, which is why he got in trouble by the IRS, right? He's like, I haven't made movies in years. They're like, what about Blade 2 and Blade 3? He's like, I haven't made movies in years. Which is why it was epic when he was in Deadpool, right?
Yes.
Okay. Anyway, Wesley Snipes was in Deadpool and Wolverine, and that was epic because they hated each other so much when Ryan Reynolds was on Blade 3 that they vowed never to work with each other. And people lost their minds. And by people, I say me and my children. Respiratory metabolic acidosis, again, as mentioned, these conditions are linked because lack of oxygen, lack of breathing, it obviously is going to cause the respiratory acidosis. And if we don't get that oxygen for long periods, we build up lactic acid. That leads to metabolic acidosis. So those two are almost always treated in unison, because even if it may not feel like it, you usually get the one following the other. Questions about ions, fluids? All right, the reproductive system. Hopefully this looks vaguely familiar because we started talking about junk yesterday, right? Bless you. Now, when we look at the reproductive system, the function is to perpetuate the species. That's the reason why we do this kind of stuff. Now, this is the only system not essential for life, right? If your heart doesn't work, you die. If your lungs don't work, you die. Your junk doesn't work, you're lonely, but it doesn't do anything for you, right? Nuns can go their entire life without ever using it, and they're okay. Some of them live to be really old. Something to think about. Is it worth it? Is it worth it? I don't know. We'll find out. But it's one of those things that you don't have to have sex to live, okay? So this is your only unselfish system because you're doing it so the species can survive, not so you can survive. So feel free to use that as a pickup line. Hey, I'm just doing this for the species. You should date me. Yeah, probably is going to work right as well as, oh, what's your sign?
Oh,
All right. So developmental aspects. We've all heard the thing that we essentially start off as female. Yes and no. Okay, you are genetically male or you are genetically female, whichever one happens to be in your DNA. In the beginning, we start off with what we call ovotestis, where they're in the position of the ovaries. They just haven't made a commitment yet. They haven't decided what team they're going to play for. Then what happens during fetal development? The baby is exposed to testosterone, and this is coming from mom because baby hasn't made testosterone yet. If you are male, you have more testosterone receptors than if you are female. So when that testosterone hits the system, it's going to stimulate the receptors on the ovotestis. We're now committed to being testes, and they're going to drop out of the body down into what will become the scrotum. At this point, we will keep the male components of the system. So you're going to keep the mesonephritic duct in place. That's going to be things like the seminal receptacle, or not seminal receptacle, that's the girl part, seminal vesicle and the prostate gland, and of course the vas deferens, and they're going to give you the boy parts. If you are female, you do not have the testosterone receptor, so you have no urge to leave the body. You commit to being ovaries, and the mesonephritic duct disintegrates. So again, the girls aren't going to have anything related to the vas deferens or the prostate gland or the seminal vesicles because they disintegrate. What we are going to get is from the paramesonephritic duct. So that's going to become the fallopian tubes, the uterus, and the vagina, which is why guys don't get those equipments, because in the guys, that disintegrates. So you can't have a uterus and testicles. You can't have a prostate gland and ovaries because the allowing of one to survive ends up killing the other one. And that's all due to those receptors. So those testosterone receptors to help disintegrate or not disintegrate those parts. And of course, that means we don't have homologs. So guys don't have anything homologous to a uterus or the fallopian tubes. Girls don't have anything homologous to the vas deferens or the prostate gland or the seminal vesicles. And again, homologous means started off the same, but now they're different. Okay. So testes and ovaries are homologous. These are our gamete-producing organs. These are our sex cell-producing organs. Just one is a boy version and one is a girl version. And of course, they are going to start to be obviously evident by the time you're born. But you'll start to see the changes in those anatomies fairly early on. So by the time you do that 5-month sonogram, it's big enough to look and see, oh, it's a boy or oh, it's a girl. So we'll start with the guys first, and I'll let them see what it's like. All right. So, of course, testes. Testes are the primary sex organ in males. Again, it is going to produce the gametes. It's going to produce the hormones that make you male. Now, they are going to be sitting inside of the scrotum, and they're going to be separated by a peritoneal raphe. So this is a thick band of connective tissue. This keeps each testicle separate from each other. And this is a safety precaution because, again, if you want to make offspring, you have to have functional gonads. And if you have a cancer or a bacterial infection wipe out both of them, you're sterile, you're useless for the population. So it's one of those, in human terms, we think everybody's worth something, but in the animal world, if you don't make babies, you just die, okay? That you're pointless in life. So we want to make sure we're not pointless, so we want to make sure that we are viable. So we're going to keep the guys completely separate, so if you have testicular cancer, it won't take out both of them. And this is why testicular cancer, it's actually very hard for it to spread, because in order for it to go from one testis to the other, it can't go sideways because the perineal raphe, if it was to spread, it would have to crawl up the vas deferens, come into the seminal vesicle and the common ejaculatory duct, go up the other side, down the vas deferens and get to the other testis, which is why when guys have testicular cancer, there's usually just one. Okay, very rarely will happen in two because they are so separated for that safety mechanism. Now, surrounding each one, we're going to have the tunica vaginalis, and as we talked about in lab, vaginalis is not the same as the vagina in the female. It's just the same Latin word that means sheath, that is wrapping around. So tunica just means a sheath. That's kind of hard to say back to back. So this is going to be sitting between the scrotum and the testis. This is basically a souvenir from the time that the testes were in the body. So of course, when they're in the position where the ovaries are, they're going to drop out. As they drop out, they pass through the peritoneum, that sac holding the digestive organs. And as they go through, they take a little bit of it with it. It's like putting your finger in saran wrap and that plastic molds to your finger and breaks off and gets stuck to your finger. So that tunica vaginalis is technically the peritoneum, the parietal peritoneum, the visceral peritoneum. The difference is now that it's not with the rest of the digestive system, we've given it a new name. So it has the same quality. It's made out of mesothelium. It produces transitate. It has a little bit of fluid, so there's no friction because, of course, friction creates heat, and heat kills sperm. So it's going to help them float around within the scrotum itself. So it does have the parietal layer that adheres to the connective tissue on the inside of the scrotum, and the visceral layer adhering to the testis itself. And in between, we've got a little bit of fluid to float around. Now, each testis is divided into lobules, and depending on the male, they'll have anywhere between 200 and 300 lobules. This is going to vary. You're never going to know unless you dissect, and most guys don't do that while they're alive because no one's that desperate to know. Now, each lobule is separated by what's called the tunica albuginea. So alba means white. So this is a dense, irregular connective tissue. Again, another barrier, another protective measure to help protect the gamete-producing cells. So it just means white tunic versus tunic. Okay. Now, within each of the lobules, you're going to have 1 to 3 tightly coiled seminiferous tubules. This is where the magic happens. This is where we're going to make the sperms. So if you were to stretch a guy out, and I imagine at some point some med student did this on a dare, but if you stretch out all 1 to 3 times 200 to 300, you'll end up with about 800 seminiferous tubules up in there. And if you stretch them all out, it's about a half a mile. So really good on Tetris packing, right? That's a whole half a mile in just one testicle. That's a lot of tubage. It looks like a brick of ramen noodles. So next time you eat ramen noodles, you're like, look at that, see? Ramen noodles. So here again, we have the tunica vaginalis, the 2 layers, the visceral layer adhering to the testis, the parietal layer adhering to the inner connective tissue of the scrotum. And here we have the tunica albuginea, the white tunic that is going to separate each of the lobules from each other and keeping the seminiferous tubules separate. Okay, so if you do eat these, so Rocky Mountain oysters, calf fries, whatever you want to call them, one of the big issues you have to do is cleaning to begin with, so that when they pop them out, they have to clean all the connective tissues on the outside. Then they have to clean off most of the tunica albuginea and the epididymis because it always adheres to it. Okay, so if you've ever prepped these for cooking, or you watch TV shows on the cooking channel, or back in the day when Andrew Zimmerman ate anything that didn't run fast enough, remember that? I mean, he was just this bald, fat chef guy that went around the world eating random weirdness. So, apparently had a good stomach. But there was one episode, they're like, look at this, we just butchered the bull, you want to eat? And he's like, so I plopped him out, cleaned him up. A lot of connective tissue cleaning because that's going to be chewy. It doesn't cook right compared to the rest of the tissue. So gotta clean those up if you happen to eat this. Okay. There they are. Doesn't that look like a brick of ramen noodles? Like you just need to add some water and a little seasoned packet. So here we have, again, the testis in its little bubble made by the tunica vaginalis, so they kind of free float in there. And we can see the tunica albuginea around the outside and separating it into the lobules. Here we have the cremaster muscle that extends up into the, oh crap, I just had a brain fart. Yeah, the spermatic. You know what I'm talking about, the spermatocord. And then, of course, we have the dartos muscle, which is directly underneath the skin. So again, the cremaster muscle is going to raise or lower. The dartos is going to pull them closer in and then drop them further out to help regulate temperature and sperm production for both of the testes. Okay. So speak of the devil, sperm production. So spermatogenesis will start at puberty. So it depends on you and your kid. So for most boys, they usually have that little squishy stage right before the puberty hits. It's like, oh, my 12-year-old just gained like 50 pounds. It's going to happen anytime, right? So they look like little marshmallow people for like 6th grade, and then all of a sudden they become tall, gangly things. If you have boys, you know what I'm talking about. The time is near when they get the squishy stage. So it's like, oh, my kid just gained some weight. Prepare. Okay. Don't have sex. So it's one of those things that where you have to reach the right level of fat. We talk about it more in detail with females because females and their reproductive cycles are more tied to fat than males are, but you do still have to reach the right body fat content for males to hit puberty as well. So skinny males are going to hit puberty later than squishy males when you compare the two. So it may be 12, it may be 13, maybe 14, depending on you and your kid and squishy levels. Now, when we look at spermatogenesis, we have three different things going on. We have mitosis. The mitosis is just normal cell division. Mitosis is going to occur in the stem cells that will produce the sperm. These are called the spermatogonium. Then we have meiosis. Meiosis is a two-step process or two-phase process that gets us from being diploid, our 46 chromosome count, to getting us to be haploid, 23 chromosome count. That way they can find an egg and make a full diploid little individual. And then spermiogenesis proper is turning it into a sperm, because at the end of meiosis, it's a beach ball. And beach balls are not very good at directing, right? All right, put one on the water, try to make it go somewhere straight. Doesn't work out too well. You got to lighten up the load because the first sperm wins, everybody else dies, right? No ribbons for second place with little spermies. So they are going to be built for speed. They have to lose most of their cell content. They have to grow a tail so they can swim, and that's all going to happen during spermiogenesis. So those three-step processes going through mitosis, going through meiosis, and going through spermiogenesis, start to finish takes about 9 weeks. So once your kid hits puberty, he's not instantly fertile. It's going to take at least 9 weeks to make sperm. Then the sperm got to mature, learn how to swim, all that kind of stuff. So you got a little bit more time after that as well. Now, of course, as mentioned, takes place in the seminiferous tubules. Adult males produce about 1 billion a day. So it's them and McDonald's on serving burgers. Okay, so that's a lot of sperm. Sperm are very cheap. This is why males are expendable, okay? Which is why traditionally we like to send them into the army so half of them go away. Okay? You don't see a lot of women going into the army traditionally because we want to protect those guys. They make an egg a month compared to 1 billion a day. Limiting issues. Okay, so that's why guys fight for females. Very rarely do females fight for guys. You shouldn't fight for guys because they're so cheap, so much sperm. And so, gotta go where the needs are. So production and survival of sperm, again, requires temperatures lower than body temperature. So we're looking at 94 degrees Fahrenheit, where again, our body temperature is usually 98.6. So you're looking at more than 4 degrees lower. So that's significantly lower than body temperature when we look at body functionality. And of course, this is why they're out of the body and in the scrotum. So we can have that cremaster muscle, the dartos muscle. If it gets a little hot, they can relax, dropping them further away from the body so we can have air circulating. If they get cold, they can bring them closer and warm them back up. Bless you. Allergic to cremaster muscle. Okay. So again, we have to make sure we have the temperature right or productions are not going to function properly. One of the biggest things, if guys have fertility problems, one of the first things they'll tell the guys is to go without underwear, right? Go commando so they can freely move and have air circulate and drop temperature. If that's not enough, they make air-conditioned underwear for guys to cool and chill the testicles to the right temperature for sperm production. I think they should make underwear like that available for anybody who has hot flashes in the summertime. Like, don't mind me, I just got my air conditioner underpants on. But it's a medical device for the guys. Go figure. Okay. So here we have starts to ends. We are going to go through our meiosis 1. That is going to take us from our diploid 46 chromosome to our haploid 23 chromosome count. And then we go through meiosis 2 that further divides out the homologous pairs. And then, of course, now we got to turn them into little spermies. A little bit better picture. So again, the mitosis is going to occur in the spermatogonium. Anytime we see gonium, these are the stem cells. These are the guys going through mitosis. Then we're going to commit to go through meiosis. So our primary spermatocyte that is diploid is going to go through meiosis 1, which is why it's the primary spermatocyte. In order to go through meiosis 1, he first has to make a tetrad. He doubles. So it looks like a little octopus hanging out there instead of an X. Once we go through our meiosis 1, that separates the tetrads from each other. That effectively makes the primary spermatocyte turn into the secondary spermatocyte that is now haploid. Then we're going to go through meiosis 2, and meiosis 2 is going to separate the genetic material of the secondary spermatocytes. And at the end of meiosis 2, we are now spermatids. We are haploid, we have half of the homologous pair, we're ready to go genetically. But unfortunately, as mentioned, beach balls don't have very good maneuverability. We got to turn it into a sperm so we can have purpose and forward motion and speed. So then we go through spermiogenesis, where we lose almost all of the organelles. We're going to keep mitochondria to help fuel the swim. We're going to keep the DNA because, nucleus is kind of a big deal, but everybody else we lose. We're going to lose a lot of cytoplasm because, again, we want to have a speedboat. We don't want to have a tugboat. And of course, we're going to put a tail on it so it can swim fast. Okay? And this is just showing you those processes. When we look at the seminiferous tubule, all of the components of the mitosis and meiosis actually happens within the tissue, within the walls of the seminiferous tubule. And as they continue their process, they're going to get closer and closer to the drop-off point. It's kind of like backing out your car out of the driveway. As long as it's in the driveway, it's not on the road and anybody can back up your car, right? You want to be that crazy neighbor, your 10-year-old can back your car up to the end of your driveway. Because it's your driveway. You can do whatever you want to it. It's not public property. Once you're on the street, now it's public property. So that's the lumen of the seminiferous tubule. Once the sperms are sperms, they drop off here, just like once your car is ready to go, it's on the street. And from there, they're going to be flushed out of the testes. The interstitial cells, they don't show them very well, but they're the cells between the seminiferous tubules. So that's why they're called interstitial cells. These guys are responsible for making our hormones, making our androgens. So here we have a complete division of labor. So production rate of sperm does not affect testosterone production, and testosterone production does not affect sperm counts. So you can have normal testosterone levels and have low sperm counts, or vice versa. So they're not tied to each other like it is in females. So we keep them separated. So testosterone, of course, is our primary androgen. Again, andro meaning male. So if you have an android, you have a male robot. So if you have an android phone, you have a boy phone, mazel tov. Okay? If it was a girl, it would be called a gynoid, which sounds creepier. So testosterone is our big player. Now, there's different types of testosterone. We're not going to get into the different types because I don't want to can't make me. Okay? But if you do care, take an endocrine class where you'll be all up in there talking about those. Now, of course, just like we don't start producing sperm until we hit puberty, we don't start producing testosterone until we hit puberty. And they're due to those gonadotropic hormones that we learned about way back on test 1. Remember test 1? Hopes and dreams, that kind of stuff. If you don't remember test 1, you've got until next Thursday. I know, that word's coming. So again, follicle stimulating hormone will help with the production of sperm. So the more FSH a male produces, the more sperm he produces. The interstitial cell stimulating hormone will help with the production of testosterone. So if we need more testosterone, you'll release more interstitial cell stimulating hormone. And this is based on testosterone production to keep it at the right levels. This is why if guys get on testosterone supplements, steroids, because they are stupid, those steroids are going to prevent the production of ICSH, which then is going to stop the production of your testosterone, which is why guys, when they're on steroids, when they come off, have bottomed out on their testosterone levels. So we will usually see shrinkage in the testicles. Basically, the interstitial cells aren't functioning, so they're shrinking a little. So here's the communication back and forth, which I find ironic that they look very similar. Okay, so the pituitary gland is going to say, hey, man, make some sperms. And if we make too much, it'll say, stop that. It will have the pituitary gland again say, make some testosterone. If we make too much, it'll say, stop that. Okay, so they're going to constantly stimulate or inhibit to make sure we have the right amount of sperm production and the right amount of testosterone. Because too much or too little is pointless for the functionality. We want to have just the right levels. The duct system. So once we have produced these sperms, they're going to be flushed into the seminiferous tubules. Those seminiferous tubules are going to join to form a network of ducts within the testis called the ret testis. It's similar to all of the drains in your house, whether they're sink drains or shower drains or toilet drains, all go into 1 or 2 pipes before they leave your house. Okay, so everyone's collecting down together. So from the ret testis within the testicle, we will leave the testis and go into the adjacent organ known as the epididymis. epi because it sits on top of the testis on the outside, kind of looming over them. So your epididymis is a tightly coiled structure because we want to get as much stuff packed into this small area as possible. If you were to stretch out the coiled ductworks of the epididymis, it would measure about 7 meters or 23 feet. So again, pretty efficient at packing all those little ductworks in one little area. It is only about 1 millimeter in diameter, so very, very thin. It's like almost a hair, a little bit thicker than a hair if you were to look at it in cross section. This is where sperm mature. This is where they go to learn how to swim. So if you were to extract sperm directly from the testis, it would just sit there and look at you in the petri dish. It would not find the egg. It would not be able to swim because it doesn't know how to do that yet. It has to mature in the epididymis. And this is where we're going to influence the sperm with testosterone. So as we've mentioned back with the hormones, when we make a hormone, it doesn't target self, it targets somebody else. So the testosterone produced by the testis doesn't target the testis, it targets the epididymis to say, make my guys learn how to swim. So that epididymis is basically a swim school, so they can mature and start becoming activated. So it takes about 20 days to move from the head to the tail of the epididymis. So if you look at production rates, to get viable sperm, it takes 9 weeks of work in the testis plus 20 days of maturing before it can swim and go fertilize an egg. So over the first 10 to 14 day period, while it's being bombarded with that testosterone, their motility is going to increase and they become fertile. So they're fertile about day 14, but they still have only made it about halfway through the organ. So they still got to get to the exit port there. Now, of course, we're not releasing sperm 24/7. So if we're not releasing sperm, the epididymis can store the sperm until it's time to release. So it depends on the male and obviously how often. So it will store the sperm. It will also help propel the sperm. When we're sending the sperm out, we don't want the sperm to have to swim out because then they'll be too tired to do their job to get to the egg. We want to help them as much as possible. Again, it's just like a water slide. That water slide is going to push you out. You don't swim down the water slide and then go into the pool, right?
Right.
Right. Watch water slides. No one's swimming down those things. They just ride there until they hit the water and then they swim. That sperm does the same thing. They just kind of hang out in the water slide until they get to the drop-off point and then they scurry off and go do their thing. So they're going to have peristaltic contractions all the way around to help. So peristaltic contractions of the epididymis, of the vas deferens to get them through the common ejaculatory duct. Then we add the water, we add the seminal fluids, and then it's going to flush the system and allow them to be dropped off where they need to go. So they can remain in storage in the epididymis for at least a month to several months. Now, the longer they sit there, they're not as fresh as they used to be, just like the longer anything sits there, it's not as fresh as they used to be. And eventually, they're not going to be efficient at swimming. And of course, we don't want to send a bunch of duds out there, so they're going to be recycled back into the system. So there's a lot of macrophages that hang out around the epididymis that eat the sperm. So if the sperm can't swim fast enough, a macrophage will tag and bag it. It's kind of an extra incentive to get out of the way because there's somebody there going to eat you if you're not ready to go, if you're not fast enough. So only the healthy sperm, hopefully, are being sent out because we don't want defective sperm being sent out. So there's our ductwork. So again, there's the ret testis, still a part of the testes. That will then dump into the epididymis, and we will journey through the epididymis over the next 20 days to mature and ready to go out through the ductwork. Okay. So the epididymis is going to connect to the vas deferens, also called the ductus deferens. Again, vas because it's big. It's a big tube compared to the previous tubes. And of course, ductus because it's a duct. Okay? And deferens because it's leaving the area. So these guys are about 18 inches or 45 centimeters long. They go up from the scrotum as a part of the spermatocord, loop up around the backside, and hook up to the seminal vesicles at the back of the bladder. They also are going to store sperm. So when a male ejaculates, he doesn't release until dry. It's a conveyor belt of sperm. So if we have little sperm A and he makes it halfway up the epididymis and halfway up the vas deferens, he's just going to hang out there until the next shoot. Okay, so we have just this continuous release and production of that sperm. So once the male has ejaculated once, the vas deferens is always going to be full of sperm ready to go, just like a conveyor belt through the system. Okay? And they, again, can also do peristaltic contractions to push the sperm towards the common ejaculatory duct, towards the urethra, so they can exit the body. Now, if males opt for birth control, one of the easiest ways males can have birth control is through a vasectomy. And again, tomy means to cut, and vas is a vasectomy is going to cut the vas deferens. So we cut and tie it off. Now, how it is cut and tied off will depend on the doctor, his personal choices, and sometimes the patient's personal choices. Most of the time, they don't just cut and leave anymore, because if you have the flappy pieces accidentally fused back together, it can reconnect itself, and then you're not shooting blanks anymore, and you can impregnate a female. And that has happened in the past, where you've had a guy who's had a vasectomy who has children because they didn't do a big gap. Nowadays, they usually remove a section, so about a centimeter worth of the vas deferens. That way, the two pieces are really far apart, so the only way they can reconnect is pull them together, which is kind of hard to do with the way the testis, sit and everything like that. And they usually cauterize them now, because if they cauterize, it burns and causes scar tissue, and you're not going to have any problems that way. Now, vasectomies, they do have reversible ones. Over in Europe, they have a chemical vasectomy. They basically shoot biological superglue into the vas deferens to clog them so the sperm can't get out. And then whenever the guy decides he wants to have kids, they go and they give them the glue be gone, basically, and it dissolves the body glue and it opens the ducts back up. Over here, instead of that, we have little plastic clips. So they remind me of binder clips, but you can go and clip the vas deferens to prevent the sperm from swimming out. And then when the male decides he wants to have children, he gets his clips removed. Actually, I have a student, he's like, I've got the clips. I'm like, thanks for sharing. They make condoms for that. He's like, I just want to make sure, really make sure. It's like, paranoid about someone getting your money, aren't you? But he's got the clips, and the only downside with the clips, they're plastic. So usually the doctors will say, don't hit these or they could break and you'll be viable. And of course, most guys are like, yeah, I'm not going to protect that anyway. So, good on you. But the plastic clips are a way to have a temporary vasectomy if you don't want to get the females pregnant and all that fun jazz. Yes, ma'am?
Is it painful since they can't release the sperm?
Nope.
And also another question: will they feel the same when they have an orgasm because they're not finishing?
No, they finish. It's just fluid, no swims.
Oh.
So you release the same amount of fluid, you just don't have any sperm in that fluid. And there's not a checkpoint on the way out going 5 sperm, 6 sperm. You feel great because 7 went by. It's off the same amount of fluid. So when guys have the vasectomy, one, it's not traumatic. I guess it would be emotionally traumatic for some guys if they're not, wieners. But it's not bad as it could be if you were a female. So if you are male, it's an hour procedure and half of it is shaving. Okay, so you go in and they give you a Valium. They spike the orange juice so you feel okay about it. They let you sit down and then their nurse comes and shaves you to make sure they can find all the bits and pieces. And then they give you the local numbing agent. The doctor will feel for your vas deferens and they'll pinch. They've got little plastic clips on the outside. They do a little incision, usually less than a centimeter. They pull the vas deferens out, use the clamps, cut it off, cauterize, stick it back in, and then the pieces on the thing. It's about, it looks like Kraft macaroni and cheese after it's been cooked before the cheese is added to it. That's about the size and consistency. And then, of course, they just do a couple of stitches and then they move on to the other side. So again, you're conscious the entire time. It is kind of funny if you're drugged up because you're not always coherent. I got to watch, at the time he was my husband, but now he's married. But I got to watch. I didn't even have to glove up or mask or anything. They're like, look at you. Cut it out. Yeah, do it. So they're like, you want to touch one? I'm like, I do. So got to look at them. That's how I know they look like macaroni and cheese. Just randomly talking about pasta throughout the day. Ironically, apparently I am. Ramen noodles, macaroni and cheese. Could be worse. I could have said it was like the rigatoni or something. Terrifying. But anywho, it doesn't take very long. They put you in a cup so you don't swell. Because if you don't wear the cup for the first couple of days, they could double in size and then your pants are going to fit weird. And then just tell you to ice. You'll be fine. Okay? Now, unfortunately, sometimes guys are double-barreled, okay? So some males may have 2 vas deferens instead of just 1. Usually it's on one side or the other, but if you're really special, you get double dose on both. So that's why they have to check to make sure the procedure went well. So about 6 weeks after the procedure, you go and ejaculate in a cup and they see if you got any swimmers. If you have any swimmers, they're like, come back in 2 weeks, you need to do some stuff to clear the ductwork, okay? So obviously. And then they'll check again. If you still have viable numbers, they're like, oh, you're double barreled, you got to come back in for a second vasectomy. I think you get a discount for the second. You should. Okay. But again, it just depends on you. Now, as mentioned, because we've got the ductwork all filled with sperm, depending on where the vasectomy happens, you may have more ejaculations to clear out of the residual ductwork there. So it may take about 12 to 15 ejaculations to clear all those collecting ducts. So you're not instantly infertile. It's going to take a little while to get through the system. Until your doctor clears you, you could still have children in that kind of scenario. Okay. So again, with he looks all smothered. So there we have the vas deferens coming up and looping around the backside and hooking up to the common ejaculatory duct. Okay. So if we have the vasectomy, again, they'll just cut here. Everybody's conscious, takes a couple hours. The equivalent would be the tube tying for the female. And if you do tube ties, the procedure obviously is surgery. So it is 6 to 8 week heal time where you're not supposed to do heavy lifting, all that kind of fun jazz. They have to put you underneath general anesthesia. So it's surgery. You have all the risks of surgeries. But they essentially do the same thing. They cut off and tie off the oviduct so the egg can't get to where it needs to go. So cauterization, that kind of scenario. They can do tube tying during delivery. So if you're on your second or more child, they'll be like, hey, while we're in there, we can just stitch something up and tie it off and you won't have any more kids. It's an option. And of course, since everything's stretched out after the kid falls out, it's a lot roomier and easier to do that. They don't have to have surgery. And you're already numbed up pregnancy and the birthing thing. So they can do tube ties while you do delivery if you want to prevent children if this is your second or third or fourth. So depending on your doctor and options, that might be a thing. But other than that, it's surgery, recovery time, repair time, a lot more traumatic on the body than a vasectomy is. And harder to reverse compared to the males as well. So the duct system, of course, once we have the vas deferens dump into the common ejaculatory duct with the seminal vesicles, we're going to dump into the urethra. That little guy is going to dump into the prosthetic urethra directly below at that border between the membranous and the prosthetic urethra. So of course, prosthetic urethra is surrounded by the prostate. Then we have the membranous urethra that's going to have the external urethral sphincter. This is the sphincter you have to have command over to prevent ejaculation. So if a male is trying to prolong sexual experiences, he has to make sure he has a good sphincter to close that off, just like he would if he wanted to make sure he didn't wet his pants. So same muscle control, same activity during that. And then of course, once that releases, then you can ejaculate from that point on. And then of course, below that, you have the penile urethra that whose job is to drop off the seminal fluid at the female. Questions about making the sperm and getting them out of the body? So of course, as we mentioned, you don't want to go down a water slide without the water. You've got to have seminal fluid to flush the system. And I forgot to mention, when you tie off, you still make sperm throughout your entire life. So it doesn't make you less of a man. Your macrophages just eat your sperm, but you are still making 1 billion a day. You just recycle 1 billion each day. So our accessory glands, these are our secondary sex organs for the males. So our first ones we have are the seminal vesicles. And the seminal vesicles is where seminal fluid gets its name from, because most of the fluid comes from these two little organs. They're going to be on the back side of the bladder, kind of stuck to the back of the bladder. They're going to secrete an alkaline fluid. Most of the male fluids are alkaline because the vagina is extremely acidic. It's not stomach, but it's almost. It's a 4 compared to the stomach, which is the 3.5 to 1.5. So it's enough to kill a lot of stuff. So we've got to have something to neutralize the acids, so when the sperm land, they don't die instantly and get burned alive. It is going to produce a viscous fluid that contains fructose, prostaglandins, absorbic acid, and fibrinogen. So again, it's going to be alkaline to neutralize the acidic nature of the female tract, which would inactivate and kill the sperm almost instantly, because the vagina is a very hostile environment. If you don't believe me, watch enough commercials about female products. So much problems, right? a yeast, it's a bacteria, it's an acid, it eats things, it's horrible. Okay, so the fructose, that's sugar, okay? Sperm are built for speed. They're so well built for speed, they didn't pack a lunch. So in order to have sugar, so they can fuel production of ATP and the swimming upstream, they've got to have sugar within that fluid. Okay, so that fructose is going to be there for an energy source to nourish the sperm, promoting fertilizing capability. So the more sugar, the faster the sperm can swim. We're going to have progestaglandins. Progestaglandins are going to work twofold. One, again, these are little local hormones, and this is going to help with sperm motility, viability, to basically tell the sperm to swim faster. Now, these progestaglandins can also stimulate muscular contractions in the vagina because, again, it's a hostile environment. We want the sperm to get to the uterus as fast as possible, which is alkaline, which is safer for them. So to help with that, you may have some vaginal contractions to help along. This is why some women will almost have an orgasm directly after the guy ejaculates. And that's because that progestaglandin is going to kickstart some contractions in the vagina, which could kickstart an orgasm within the female. So sometimes ejaculation will help the female to end her process. Sometimes it doesn't. It just depends on the female mood, all issues that can lead to those things. Fibrinogen. Fibrinogen makes a clot, okay, just like it does in your bloodstream. We're going to have a clot here. So again, why do we think we need a clot going on? So it doesn't fall out. Again, it's going to basically stop up things. So when the female stands up, nothing falls out because sperm on the floor can't fertilize an egg, okay? Not unless you do something disturbing. So again, to help promote fertilizing capabilities, if women are trying to get pregnant, that's when the doctor will say things like you need to stay laying down, you need to tilt your hips up so everything kind of gravity feeds to where it needs to go. Don't get up for the first 10 or 15 minutes after the drop off. That way those sperm can get to where they need to go. It has even been hypothesized that the reason women have orgasms is to create those contractions that draw sperm in. So you'll have some scientists that say the only reason women get orgasms is so they can get pregnant. Now, having orgasms does increase your chance of pregnancy. So if you want to get pregnant, have an orgasm. I know if it was only that easy, but it will help with those muscle contractions. And what will also help is to prevent things from falling out. This also helps with the competitive edge. Now, hopefully humans aren't having the same issues that dogs have, right? We've seen what happens when dogs go into heat. There's a line, okay? Hopefully girls don't have a line. Yeah, disturbing. So this sperm plug, other than keeping sperm from falling out, it actually makes a clog that prevents other sperm from getting past. So if you have one male drop off, and then you have another male come and drop off after, his sperm is going to try to swim up, and it's going to hit the roadblock, and it's not going to get to where it needs to go. So that clog not only helps the sperm to not fall out, it helps that sperm to be more successful because it gives them a edge, because the next sperm drop off can't get to where it needs to go within about 10 or 15 minutes. So they get to swim upstream a little faster, so it promotes their fertility. So again, hopefully in humans, that's not a big deal. But again, for dogs and stuff like that, it is. If you are a male and you're third in line, you still want to have that chance. So when you get to animals, they actually have eating sperm. So they'll have some sperm that their job isn't to fertilize, it's to eat the sperm plug so the next sperm can get through. So there's a lot of arms race with sperm and all that kind of stuff in males out in the animal world. They haven't observed that in humans yet because it hasn't been that desperate in fertilization, right? Unless you're on that chick that's in the Maury shows. Remember those? Fifth guy in, he is not your baby daddy, and then she all freaks out and falls on herself. It's like, dude, fifth guy in, calm yourself. Good luck. You're not supposed to be able to be that big of an issue. But again, if you go into breeding in animals, sperm plugs are very big deal, helps with the isolation of the sperm gas. Okay? So fluid secreted by the seminal vesicles makes up 60% of the seminal fluid. Again, this is why it's called seminal fluid, because it's named after the seminal vesicle. Now the vesicles merge with the vas deferens, forming the common ejaculatory duct. That way the fluid and the sperm, the swimmer and the water in the water slide hit at the same time to prevent any kind of damage or injury to the sperms as they go out the system. So there's our seminal vesicles, there's our vas deferens joined to form the common ejaculatory duct that's going to enter into the prostatic urethra. Think that your questions so far?
No.
Excited? Or disturbed?
All of the above.
Yeah, that's my job. All right. We got 2 minutes. We can either rush the prostate, or we can slow things down in prostate tomorrow.
Rush the
Everyone's
prostate.
like, I don't want a prostate at all. Prostate tomorrow. It'll be like we're proctologists. Fun fact, proct is Latin for anus. So if you're a proctologist, you're actually an anus doctor. Yeah. Things that people don't tell their parents when they grow up. Let's go to lab, where we're going to do something crazy like have a bonus quiz, and then we're gonna go play with Victor and Victoria.
I think it is. Is he working today?
He ended up working last night, which is really weird. Apparently, he does not like Mr. Gonzalez. Because I got Victor to work for me last night.
Stop, bro. What? Oh my gosh. He's like, I got
You can turn it in and out.
Wait, let's walk across the street. Let's go to
No, I can smell the alcohol content in your thing.
yeah.
Best. You know it.
Oh, drink.